I feel privileged to have such a distinguished chief guest, Padma Bhushan Dr. V.K. Saraswat, who need no introduction. Dr. Saraswat is a scientist with a vast experience in defense research spanning over several decades. Formerly, he served as the Director General of the Defense Research and Development Organization and the Chief Scientific Advisor to the Indian Minister of Defense. Currently, Dr. Saraswat is the Chancellor of Jawaharlal Nehru University, New Delhi, and the President of Sri Chitira Tirunal Institute of Medical Sciences and Technology, Thiruvananthapuram. He is also a member of NITI Ayog. He has been featured in several newspaper articles related to COVID-19. It's my honor to invite Padma Bhushan Dr. V.K. Saraswat to inaugurate the symposium. We welcome you, sir.
Good afternoon to all of you, and very warm greetings to all the organizers, the esteemed guests, and also the participants and dear students who have really gathered here to discuss a very important topic, which is the host genetic response in COVID pandemic times, a boon or a bane. All of us know that in this particular scenario of COVID-19, this pandemic has become a major global public health problem with more than 200 million cases and 4.5 millions of deaths worldwide. So in this particular context, I would like to talk to you about the importance of genetics as a science in relation to the COVID-19 pandemic. Genetics is an important not only to understand the cause of a disease, but also to recognize the manner in which an individual responds to the particular therapies. Genetics are playing an important role as a science to face the global threat of COVID-19 in identification, management, treatment. Now, the contributions that genetics have had and will continue to have in the identification of the new SARS-CoV-2 is in the following way. That is development of new diagnostics and techniques, prevention of infections and development of severe symptoms, and design of vaccines, and in the proposal and evaluation of treatments. Genetics is a key tool for the development of precision medicines in the COVID-19 treatments. Although many challenges remain to be solved, it remains a branch of science that provides us with a range of options and potential to optimize and develop new therapies in turn. Until recently, it was very difficult to establish accurate genetic maps of all human diseases. But presently, with the cutting-edge technologies, the cost of genome sequencing continues to drop rapidly, with results in many more human genomes being sequenced. People react differently to drugs as one size does not fit all. Patient population having same disease phenotype can behave differently in response to the same drug. Based on the response to the drug, they can be categorized into non-responders, toxic responders, and of course the responders. So why does the drug response vary? That is the most important part. One reason is genetic variation. Primarily, two types of genetic mutation events create all forms of variations. One is a single base mutation, which substitutes one nucleotide for another, which is called SNP, as we call it colloquially these days, and insertion or deletion of one or more nucleotide, either in the tandem repeat polymorphism or the insertion deletion polymorphism. All of you know that polymorphism is a genetic variation that is observed at a frequency of more than 1% in a population. Another reason is for the variation in the drug response is the single SNPs are single base pair positions in genomic DNA, at which different sequence alternatives called alleles exist, wherein the least frequent alleles has an abundance of 1% or greater. So SNPs are the most commonly occurring genetic differences. Another reason is the tandem repeat polymorphism. Tandem repeat or variable number of tandem repeat are a very common class of polymorphism consisting of variable length of sequence motifs that are repeated in tandem in a variable copy number. Based on the size of the tandem within units, it could be microsatellites or short-term tandem repeat or mini satellites, which can make the difference. Another reason is due to the individual variations. There are more than 20 to 40% of patients benefit from an approved drug. About 70 to 80% of the drug candidates fail in clinical trials, and many approved drugs removed from the market due to the adverse drug effects. The use of DNA sequence information to measure and predict the reaction of individual to drugs is in terms of personalized drugs, also faster clinical trials and less drug side effects, which is actually nothing but the pharmacogenetics, as we call it in our technical terms, which is nothing but the study of inter-individual variation in DNA sequence related to the drug absorption or disposition and or drug action, which we call as the pharmacodynamics, including polymorphic variation in genes that encode the function of transporters, metabolizing enzymes, receptors, and other proteins. The study of how people respond differently to medicines due to their genetic inheritance is called pharmacogenetics. So correlating heritable genetic variation to drug response is important. An ultimate goal of pharmacogenetics is to understand how someone's genetic makeup determines how well a medicine works in his or her body, as well as what side effects are likely to occur. So that is the genesis for right medicine for the right patient. Now, there are determinants of drug efficacy and toxicity. A patient's response to a drug may depend on factors that can vary according to the alleles that an individual carries, including some pharmacokinetic factors like absorption, distribution, metabolism, and elimination, and the pharmacodynamic factors like target proteins and downstream messengers. Now, we have seen in this present pandemic, the way we have used mRNA vaccines for variety of actions, what we have generated so far. COVID-19 virus containing spike protein, all of us studied that, and then mRNA with instructions for making the spike protein has been developed in the lab, and then mRNA enters in the human cell through the mRNA vaccine, and COVID-19 virus spike protein is created. That proteins are recognized by the immune system, which produces specific antibodies against the COVID virus. If that individual is infected with the COVID virus, antibodies bind to the virus and stop it from replicating the COVID virus. This is the kind of genesis on which all the vaccines have been developed today. And another one is the vector vaccine, in which the COVID containing spike protein is seen. Then spike protein genetic material is extracted and inserted into the inactive virus. And these inactive harmless virus enters the human cell through the vaccine, and COVID-19 virus spike protein is created. And spike proteins are recognized by the immune system, as we have seen earlier, which produce specific antibodies against the COVID virus. And that is how the system works when it is infected. Now, human genetics helps to identify potential drug targets to start drug discovery. That is another one. The key steps to be followed are to map the genetic differences in those with disease versus healthy, and understand how these genetic differences lead to the disease. Develop drugs against these targets that reverse disease processes in the population. That is how the study is carried out. Then, what are the different approaches for drug development? One approach is the traditional approach of drug development, where lacking human data. In this approach, the gene drug target of interest was selected and administered into the small animal like mouse to carry out in vitro experiments. After successful experimentation results, the trials are conducted in human. All these activities were leading to the higher cost and lower successful rate. Whereas the new paradigm drug development mechanism is driven by human body genetic data. In this approach, the gene drug target of the interest is selected, and the proxy signal nucleotide polymorphisms, SNPs as they call it, is being used as a signal for drug efficacy and safety. Here, the target validation is guided by human data. The new approach is having higher success rate and lower cost as well. The impact of human genome project on medicine, how it is happening, it is very important. If you look at the diagnostics where you are trying to do the identification of genes for monogenic disorders, like through the molecular tests, for modifying factors through the genetic screening, for predisposing risk factors in which we need to look at the prevention of the disease, and that change normal cells to cancer cells in case of oncogenomics, for variation in drug response. And therapeutics, when you take, there is the identification of new drug targets and streamlined drug discovery and approval process, which is part of our pharmacogenomics studies. So this is how the genome projects are done in medicine. There are, of course, challenges in developing genetic medicine. And what are those challenges? One challenge is that nucleic acid delivery, which is a major problem because these acids are unstable and do not readily enter the cells. Second one is the drug product development, manufacture, and scale up, where genetic medicines are complex drug products and are traditionally difficult to develop and challenge basically for scale up. Then, of course, there's a knowledge gap, which is genetic medicines are emerging technology and new teams have the full complement of experience in developing this drug product. That is one thing what we are looking at, knowledge gap. Last is the different needs for different programs. Genetic medicines are being developed as personalized medicines for the mass administration and by teams with a wide range of capabilities. These are things which are happening today across the globe, but many of them are in isolation, many of them are synergistic. But what is important is to work towards a common cause. Now, gene therapy has the potential to treat all of the classes of disorders. For example, infectious diseases as a result of infection by a virus or a bacteria, cancers or inherited disorders like genetic deficiency of an individual gene product or genetically determined inappropriate expression of a gene, immune system disorders, which includes allergies, inflammations, and also autoimmune diseases, in which body cells are inappropriately destroyed by the immune system cells itself. There are hurdles in clinical development of gene therapy. What are those hurdles? General issues are efficiency of gene delivery. There's a major issue on this. Consistency of expression, that is the position effect, what is important. And risk of cell transformation. And another factor issue is immune response against vectors and the inserted genes. So ex vivo cell therapy, selection of the engineered cells, how do you select them? Amplification of the engineered cells, another hurdle. Then clonal competition, which also is a major issue. And in vivo, it is stability in vivo and targeting and toxicity. To end, I want to mention that where does the gene therapy or gene research is going? There are more than 2,500 gene therapy research projects that have been completed, are ongoing, or are in pre-clinical development. Continuing research is on. After 50 years of research, success stories are emerging with 2 gene therapies approved in the US. These are not even hemophilia. So this is the kind of scenario as far as overall gene therapy and gene medicine and gene is concerned. But what is important is that the US Food and Drug Administration has approved 7 cells and gene therapies since about 2017. And these drugs are like what we call as Abecma and Breyanzi and Kivria and all that. These are various drugs, 7, 8 drugs which have been approved. But even there, mostly the marketing done by the concerned agencies like Novartis, Roche, and all that. They are basically for therapeutic covering some areas like cancer. They are also covering some of them. Mostly they are related to cancer and myelomas and things like that. So this is where the entire process of gene therapy is moving. But I personally believe that there is a lot more to be done. A lot more understanding has to be created. And one thing which will play a major role in this area, I would say, is the high-end computing facilities which are available for gene sequencing today, including the supercomputers and also the drug design using the genes and all that. I think we should make use of them in its totality, work with the computer engineers, with the software engineers, and work out the major software packages for use like open source for all the researchers. That will certainly augment the kind of gene research which we are talking of. And in the context of pandemic, it has already made a major difference, but more and more understanding of the COVID and COVID-related viruses can be created if we have a concerted effort of gene-based scientific research. Thank you very much, and I wish the conference, the seminar, a great success. More ideas would come, and the science will